Hi class, we are getting into Mendelian genetics. So now we're going to start looking at where we inherited those traits from and how we determined which traits we see and which ones we don't, and why maybe a trait skipped your parents, but yet you show it. So going through things like that. So going to go through a couple key terms first. So a couple things, and we've talked about a few of these already. So knowing that a gene is going to be a specific part of the chromosomes that's responsible for the trait. So when we're talking about a gene, you're looking at your base pair sequencing. And so not just one or two base pairs, but a long kind of section of DNA that's got your A's, your T's, your C's, and your G's that we talked about in the genetic material. And so that's what's going to code for a specific trait. And then when you're looking at alleles, they are going to be the different forms of the genes. So in the idea that eye color, you could have blue eyes, green eyes, um, brown eyes. There's different shades, so that's not quite perfect with the example for this or that, but it's one of the ones that kind of people can see most generally. If you're talking about kind of what's called a widow's peak with your hairline, so if you actually look at your hairline and look at your family's hairline, you'll be able to see some of you have what's called a widow's peak, and it kind of points at the top of your hairline. And then others will have a flat one right across. And so those are going to have specific traits. And so if you're looking, the dominant trait is that widow's peak, and the recessive is that flat across. And so sometimes you'll have both the widow's peak gene allele. Sometimes you'll have the allele for, if you have the flat, you only have the flat allele. If you have the widow's peak, you could have the alleles for the widow's peak, or you could also carry the flat line, it just doesn't show. And so that's the difference with your genes and alleles. Okay, then we get into looking at specific traits. When we're actually talking about the physical actual trait, that's what's known as our phenotype. So when we're talking about the idea with the widow's peak, that's your phenotype. We're going to kind of go into some pea plants because that's one of the scientists' kind of materials that he studied with. So here you're looking at your seeds, a yellow seed, the yellow seed, a green seed, those are your phenotypes. Genotypes are actually the genes that code for it. So here we just use specific letters that relate to the trait. So you use the letters, and then you have two copies because you have two copies of every chromosome, one from mom, one from dad. And so that's why we always have to have two letters in an individual. Okay, when you're comparing your genes or your alleles, they could be either heterozygous, which means they are non-identical. So kind of if you look at the table here, you're heterozygous. And then homozygous means that they are going to be the same both capitals or both lowercase, where heterozygous is always a capital and a lower. And we'll explain what those capital and lowercase letters mean as we kind of work through the notes, so don't get too hung up on that. Okay, so who was the kind of founder and father of genetics? Gregor Mendel was the he was a monk that actually started kind of looking at pea plants and seeing how traits got passed from generation to generation. And so what he was able to do is he was just able to study the specific pea plants because they had certain traits that never changed generation to generation. And kind of from there, he was able to kind of introduce a little bit more about inheritance. That being said, it really wasn't widely accepted when he was doing his research as it is now. So a couple of things he was able to discover while he was doing his experiments is that pea plants have two contrasting characteristics. So if you kind of look at the table on your slide, you'll see for stem length, it was short or tall. There's no medium trait. Same thing with your pea pods, it was inflated or pinched. It was green or yellow. There's no third, fourth, fifth trait. So it really made it easier to study and understand inheritance because there's only two traits. They were true breeding plants, which meant generation after generation, those traits didn't change. They stayed the same. And that they were a closed population, which means that he could control the pollen to another flower and allow for fertilization to happen. So he had a lot of ability to control what the parents' phenotypes were, and then seeing what the offspring's phenotype would be too. Okay, so his first experiments he bred were pure for the traits. So what that means is that when you bred them, all the traits, all the offsprings would be the exact. So if you took yellow and you cross with the yellow, it means every single offspring would be yellow. If you took a green with a pod green and a pea, and the offsprings would all be yellow. That's what it's meant by pure yellow and pure green, is that all of the offsprings would only show the same trait. So that's what he used first. Okay, what he was able to determine is that you started off with this pure, okay? And so when they say pure, the one thing we haven't talked about is the genotype. So these are the letters that correspond to the alleles. So the yellows pure means that they are going to be homozygous. Y's are kind of hard because you have to really make sure that it shows the difference between uppercase and lower cases. Okay, so I kind of do printing and then a cursive Y to help figure out the difference. So make sure you realize that's what it means by pure, is that they are going to be homozygous. The offsprings were all hybrid. So that means that they are all heterozygous for the traits. Okay, so we had a green one. What happened to the green? So it wasn't that the green pea plant just kind of disappeared. It was that the yellow is dominant, so it hides that trait. So then if you were to cross two F1 generations, what do you think might happen? So this is his kind of result of his first experience. And so again, remember, the P generation means they're pure, so they're homozygous. F1 would be your heterozygous. And then from there, he's going to take two F1 generations. So they end up being, where with my two parents are going to be heterozygous. And then all of a sudden, that green trait popped up again. And the reason the green trait popped up is because some of the offsprings got both of the recessive alleles. And so when we're talking about dominant and recessive, what that means is if you carry the allele for the dominant trait, you only see the dominant phenotype. If you carry both recessive alleles, you will show the recessive trait. So you can only show the recessive trait if you show both of the recessive alleles. So just kind of be aware with the difference of dominant. Dominant, you only need one capital letter. Recessive, you need both lowercase letters. And we'll kind of work through that a little bit more when we're going through it. Okay, so from this experiment, Gregor Mendel was able to come up with what's called the law of segregation. And so the idea is, how did we determine which offspring got those alleles? How did they come up? And so it's all based on meiosis and how you separated those different genes. And so if you were looking for the yellow pea plant here, grab the pen instead of the highlighter, guys. Okay. The gametes can only have capital Y's to give to their offsprings, where the lowercase, again, can only give lowercase y's. So this is why 100% of their offsprings were heterozygous. But if you think of the heterozygous, they have the option for their gametes to have either the capital Y or the lowercase y. Totally depends at random which one it has. It's literally like putting them in a bag and pulling them out randomly, and the probability is based just on randomness of picking it. And so that the probability that you ended up with your traits was there's no rhyme or reason. It was completely random the way meiosis separated those genes. And it has to do a lot with, if we remember, your independent and random assortment and how they lined up in meiosis 1 at your metaphase 1. Okay, so let's do a quick check. These aren't actually in your slides. So as you're reading through the questions, hit pause, read through it, try to answer it. Unpause. We'll kind of do these throughout the next couple notes as well, just to kind of make sure you're understanding everything, and then we'll talk about them in class as well. Okay, so I'll give you a minute to read the question and then answer it. Okay, so which of the following genotypes would code for recessive phenotype? So remember, genotypes are the actual letters. Phenotypes is your traits, so make sure you know the difference. And so because it's recessive, we said it has to be both lowercase for you to see the recessive trait. Okay, all right, let's try the next one. Jess has brown hair. This is an example of phenotype because it's the actual physical characteristic. Okay, we're going to keep going. Okay, so The next one, in pea plants, yellow seeds are dominant over green seeds. What would be possible for the plants to see the yellow seeds? If you dominate, you only need one capital letter. So D would be the correct answer because it could be this or that. Okay, so now we're going to look at how to kind of practice a little bit more with coming up with your genotypes. So when you're looking at your genotypes, say we're talking about the height of the pea plant, it can only be tall or short. Remember that because tall is dominant, short is recessive. Tall only needs one capital letter, where short will need two lowercase. So let's figure out, based on your law of segregation, kind of how to split these different genotypes up. So how many different gametes is it possible for the plant with the genotype T? So if you're looking, it has the option to this or this. And since it's the same letter, all gametes will all have capital T, which means all of the offsprings are going to be tall. Doesn't matter what the other parent is, all the offsprings have to be tall based on this one. For here, the gametes could be capital T or lowercase t. So now the other parents' genotypes matter as well. Okay, hopefully this is going okay. We're going to go through it again in class as well, so just so you're kind of aware. Okay, so now we're actually going to get into how to solve these genetic crosses. And what we use for genetic crosses is these Punnett square, and we're going to use it for the next like 4 or 5 lessons, I think. Once you get the hang of it, they're pretty repetitive. It's just making sure you understand kind of the basic concepts of it. Okay, so let's go through and work through the first example. In humans, brown eyes are dominant over blue eyes. So what that means is when you're doing it, brown eyes has to have one capital B, blue eyes would have to have the lowercase b. So then it says a man is homozygous dominant for brown and the woman is blue eyes. What would their child be? So going through and figuring it out. So we're going to work through step by step. So first thing we need to do is write out the dominant trait and recessive trait. So capital B, as I kind of said already in the other one, is going to code for brown eyes. And lowercase will code for blue eyes. Okay, then we have to figure out what dad is and mom. So remember in the question, dad is homozygous dominant. So homozygous means you have both the same alleles, and mom has blue eyes. So since she has blue eyes, she has to have both recessive traits or else she wouldn't have the blue trait. Okay, so then you need to draw your Punnett squares. This one's kind of done by the computer. I like to have the extra lines in there just to separate. So when you're doing your Punnett square, you kind of have a little square like that on the side. Then you're going to add in your gametes. So you have to figure out what the gametes can be from the parents. And since dad can only give uppercase B's, we'll put dad on the top. It doesn't matter if you did dad on the top or dad along the side and mom along the top. So then you have to separate out your gametes. And so that's why I kind of sometimes put the extra lines in. Okay? And so that's how you're going to separate it because dad, we know from the other side, was 2 uppercase B's, mom was 2 lowercase B's. So your uppercase B's get separated, the lowercase B's get separated. Now what you're going to do is kind of almost like a drag and drop thing. So this B is going to go into this square. This B goes to this square, so you'd be uppercase and a lowercase. And then same thing, this B is going to go all the way down here, and this little B goes over here. This B goes down here, and a little bit. What I always generally tend to do when I'm doing this is I put if there's a dominant allele, I put that first. So you could have written it like this in some of the squares. I just like to make my life easier and always put the dominant letter first. So that way when I'm looking at which ones are showing the dominant trait, it's easy for me to kind of pick out. So this is step 4, filling it out. Now we're going to write the phenotype and the genotype ratios. So if you look, every single square is the exact same. So that means 100% of the offsprings are going to have the exact same allele, uppercase, lowercase. And if we remember, because brown eyes is dominant, they only need one B. So 100% of their children will have brown eyes. Okay, we're going to go through another example, so don't worry too much. And we're going to do a couple checks and go from there. Okay, so let's go through another one. Okay, so it says a woman and man are both heterozygous for eye color. Okay, so eye color in humans have lots of variations. We're going to pretend it's just brown or blue just to make things easy for us. Okay, so it means mom and dad are both heterozygous. So they're both brown, we're assuming, because we're assuming it's just brown or blue eyes. Okay? We're not forgetting all about you green eyes. We're just not going to use those eye colors right now. Okay? So those are mom and dad's genotypes because it's heterozygous. Then we are going to fill in our chart. I like to just have the extra ones there. Just personal preference. Okay? So parent 1 on the top, parent 2 at the side, and you know all the gametes, so based on the law of segregation, you have to separate them. Then you're going to do your drag down, drag across. So this square is going to have both capital letters. Then we're going to take this B and drop it down here in the lowercase and bring it over. And then you can drop, remember I always said I like to do the uppercase first, but that's just preference. Okay, so now the Punnett square is completely filled in. From there, I need to figure out my genotypes. And when we're talking genotypes, we're talking about the offspring, so the babies. Okay, so you're going to have 1 for a homozygous brown. Then you're going to have 2, so you have this guy here, I'm going to have this, and this guy here that are the exact same, so that's where the 2 comes from. And then the last one is the lowercase ones here, so 1. And if we remember, for our phenotypes, you're talking about the actual physical characteristics. So these are 3, uppercase, lowercase. Oh, sorry, I'm writing the genotypes, not the phenotypes. Okay, so these are going to be for brown and 1 blue. And so that's how you're going to answer the question. So then it says, what are the chances the offsprings would be brown eyes? You would have a 75% chance. And with your blue eyes, you would have 25% chance. Because it's 3 out of 4 or 1 out of 4. Okay? All right. So we're going to look at a couple little tricks on how to check your crosses and know a little bit about some stuff. Okay? So when you're looking at kind of some tricks to how to figure this out. When you're looking at your pure parents or true breeding ones, they're always going to be homozygous for that trait. Okay? So that means that they're always going to show those, remember we said the capital letters. Or 2 recessive letter, depending if it's the recessive. And these are what's called your P generation, so your parent generation. The offsprings are all going to be showing the dominant trait. So your F1 generations are always going to be these genotypes, and they're always going to show the dominant trait. Those are what's called your F1 generations. Okay? And then when you cross 2 F1 generations, so that means that you have 2 with the heterozygous alleles, you're always going to end up with this 3-to-1 ratio. It's never going to change based on 2 heterozygous. If I had did a heterozygous and a homozygous parents, the ratio doesn't work. It only works if you are crossing, say, what your F1 generations. So what the F1 generations means you're crossing 2 heterozygous parents. So make sure you're aware of it. It's always going to be for your F1. I'm going to just write a little note down here. 2 heterozygous parents. Then you will always see this 3-to-1 phenotypic ratio, and again, it's your 3-to-1 or the 3-to-1 phenotypic ratio, and then the 1 to 2-to-1 genotypic ratio. Okay, so just be aware that that only works if both parents are heterozygous. If you're doing your true breeding where your both parents are homozygous for the opposite trait, 100% of your offsprings are going to be heterozygous. So just kind of be aware there's a little bit of tricks to know for that. Okay, and the very last topic of this note is your test cross. So we don't always know the genotype of the offspring. We can always see the phenotype, but we can't always tell the genotype. And so how do we figure out the genotype of that, say, species we're looking at? We can perform what's called a test cross. So you have an unknown genotype, you always cross it with a recessive. By doing so, you'll be able to see what traits pop up. And we'll go through an example so you can kind of understand where that's coming from. Okay, so there's a little example here. It says, in hamsters, rough coat is dominant over smooth coat. Okay, you have a hamster with a rough coat. So that means it's this, but we don't know this second letter. So it could be homozygous dominant or heterozygous dominant. We don't know. So what we need to do is we are going to do it with a test cross. So let's practice our test cross. So that means that we know one parent is something. We still don't know what the second allele is, but they have one cap. We're going to cross it with a recessive parent. So I always put the recessive one on the side, just out of habit. You can put it on the top if you want. So then we know this one here, but we don't know this guy here. Okay, so does that make sense, everybody, so far? And it's okay to put a question mark for a test cross. That's what we're trying to figure out. So we're going to do the cross that we know. Okay, so then hypothetically, it could be this letter or this letter. So I like to split this just in the middle and kind of do both. This one gets to be a little bit trickier, and that's okay. We'll go through it again. Okay, so if the parent had the both dominant alleles, you can see all of their offsprings would show the dominant trait, which is the rough coat. But if it was heterozygous, you can see 50% of the offsprings would show the smooth coat. And it's okay to say you don't know because you're not actually performing these crosses. It's saying it can be this or that depending what their offsprings are, and that's an acceptable answer. So when you're looking through here, your answer could be, give me a second and I'll write it better. So okay, so that's the first half of the answer. And then the second half is if it was, I'm just going to kind of short form it because I'm running out of space, it was heterozygous, then sum of would show the smooth coat. Okay, and so that's okay for a test cross. So you would have both points. So it's not like it's a this or that answer. It's like if this happens, then this will happen. If this happens, then another thing will happen. And that's okay for an answer. Okay, we're going to do two little quick checks before we finish the note. So again, read through them, try them. They're not in your notes. It's just good little practice for you. So in pea plants, the spherical seeds are dominant over the recessive, the dented seeds. Okay, so genetic cross for two are heterozygous. So remember what we said. So we're talking about heterozygous parents. So S's are really hard. So make sure if you're using S's, it's hard. It's easy to tell which ones are the capital and lower cases because it can get confusing if you're not making it at that. Try to stay away from like S's and C's. Even X's can be hard as well. So try to stick with letters. That's really easy to distinguish between the capital and lowercase letter. All right, so then we're looking for the spherical, which is your dominant trait. So if you remember our tricks, we know that 3 out of 4 are going to have that dominant trait. If you wanted to do a little quick test cross, or you're not test cross, just a Punnett square cross, sorry. But see what I mean about S's? S's are just hard. So you can see that 3 out of the 4 are going to show that dominant trait. Okay, all right, one last little check. Okay, it says it's 2 hybrid parents. So hybrid means it's heterozygous. Okay, have the yellow seed will yield how many? Green. So if yellow seeds are dominant, so this is again, remember our little tricks. So you do your cross and you should be able to come up with your 25%. If you want to do your little cross, that's okay. It's always good practice. And you'll see just 1 out of 4. So that's where your 25% comes in. Okay. All right. There's lots of practice for you to go through. I know that was a long note, and then we're going to go through lots on teams when we get to class. Okay. Thanks, guys.